The topic will be focusing on gene editing. All right, so I know that it's very close to lunch, and you're probably wondering why. What is this image? So I asked ChatGPT to generate an image of a scientist who are very hungry, but still at the same time making CAR T cells using CRISPR, and artificial intelligence never disappoints me. All right. Okay, this is the outline of my talk. I'd like to begin with the definitions of gene therapy and gene editing. So because we're going to be talking about these terms over and over, so it's just, I think it's just better, especially for the students, to understand exactly what gene therapy and gene editing is. And then we will move on to how a gene is edited. And then we will be focusing on the CRISPR-Cas, which is one of the gene editing techniques. And then I will talk to you about the gene therapy in current medical practice and end the talk with CRISPR technology in CAR T cells. And as you can see here that I also put the picture here. By now, everyone probably know who they are. These are Dr. Charpentier and Dr. Doudna, who are awarded with the Nobel Prize on their discovery for the development of a method of genome editing, which is CRISPR to be specific. And I also put the structure of the car over here because we have, repeatedly visit this for a long time. So yes, this talk will be, focusing on these two topics together to celebrate all the scientists who are behind these wonderful, developments, including us. All right. Yes, and. Okay, the definition of the gene therapy and gene editing. So I'd like to begin with this diagram over here. Yes, so this is the central dogma of biology, as you might have seen this many, many times. So the proteins are actually translated from the information of the mRNA, which is encrypted in the gene, which is the DNA. So the DNA sends out the coding information through the mRNA and then get translated to become the protein. So whenever we think about the gene therapy and gene editing, we might want to keep in mind that it's just the beginning of the process. We might have to keep in mind what protein we get look like in the final process. So for the gene therapy is this is the definition from the Nature Site table. It says that gene therapy involves the introduction of a new genes into the cells. And the purpose is to restore or add the gene expression. Yes, as you can see here, that it's just say that it's the introduction of the new genes. So this gene or this piece of DNA, or transgene rather, yes, it could integrate into the genome or stay outside of a genome as the episomal DNA. So either way is fine. It counts as gene therapy as long as it restore or add the gene expression. But for the gene editing, the difference is the gene editing is a group of technology that gives scientists the ability to change an organism's DNA. So this is a difference between the two. The gene editing is, we purposely change the DNA sequence. All right, so gene editing is just one arm of the gene therapy. Now quickly move on to the next topic. How is a gene edited? Again, I'd like to start with this diagram, which is just a simple diagram of how the gene is repaired after the DNA double-stranded break. The upstream of the event is the double-stranded break, And then you can see that the majority, the main repair pathways are first non-homologous enjoining, and also the another pathway is HDR or homology directed repair. The difference between the two is the first one, or the NHEJ, doesn't rely on the homology, doesn't rely on the homologous chromosome. It simply just join the break ends together. It's quick and simple, and it happens most of the time in the cells, and it can happen at any time of the cell cycles. So it's usually end up with some kind of mutations, could be insertion or deletions. But if the cells decide to go the other way, to another pathway of homology-directed repair, this is the error-free repair. Why? Because the break ends will start to look for the similar sequence on its homology to find the missing information. And after that, it's going to incorporate that missing information at the missing part. So that's why this is the error-free process. But we can trick the cell into thinking that this, whatever information, whatever DNA sequence that we put in between the homology sequence are the missing part. For example, if you would like to add a GFP gene into the break site, you can just put GFP gene between the homology sequence and give it to the cells in the form of donor template. So yes, and this is some of the time the cells will get tricked into incorporating that foreign DNA sequence into its genome. And so the take home for this is that gene editing exploits the process of DNA repair that follows DNA double-strand break. Therefore, when we would like to edit a gene, we will have to induce DNA double-stranded break. And there are some tools that we can do that. For example, the zinc finger nuclease, the TALENs, or the transcription activator like effector plus nuclease, and the CRISPR-Cas9. So please keep in mind for the students, that the CRISPR-Cas9 is only there to induce the break, and whatever that follows, it's up to how you design your experiment. And then, yes, this is, if you want a specific sequence to be incorporated into your genome, you can also provide the cells with the donor template for homologous recombination. And finally, the method, we will have to deliver all of these gene editing tools into the cells. And that was the previous lecture by Dr. Doungkamchan. The methods to deliver nucleases and donor templates into the cells. All right, so I would like to focus on the first one, which is the method to induce the double-stranded break. So if we can see here, some of the methods that was mentioned previously, such as transposon and lentivirus, it can cause the gene editing, yes, but in a non-site-specific manner. It may not be entirely random, but it's non-site-specific DNA editing. But these groups of technology, this allow us to perform targeted DNA editing, meaning that we can direct the endonuclease at a specific site on the genome. And these technologies are, for example, these three. And okay, let's just look at each of them. The zinc finger nuclease and the TALENs, yes, you can see over here that there's always the component of nuclease, which is there to induce the double-stranded break. So the other component is the component that helps direct the nuclease at the specific site on the genome. The zinc finger is the protein that can bind to the DNA, but it recognizes the nucleotide the three nucleotides at a time. So it's like, if you would like to recognize this part of the, the sequence, you can just, do this combination of zinc finger protein. And then the zinc finger protein will be tethered to a FOC1, which acts as endonuclease. Likewise, for talent, instead of recognizing three at a time, it allows more flexibility by recognizing only one nucleotide at a time. But the downsides of these two approaches is that we cannot synthesize this in our lab. We will have to entirely depend on the company. Yes. And we don't like that because, it costs money. Well, I'm sorry. I know that some of you guys are from the startup. And yeah, thank you so much for being here. Okay, and now moving on to CRISPR-Cas9, right, which will be the spotlight of today. Yeah, and this is the side-by-side comparison between the natural CRISPR-Cas9 and the engineered CRISPR-Cas9. As you can see here that, again, the Cas9 acts as the endonuclease, which is the important components of both systems. But the direct, the system that directs the Cas9 to specific place in the genome for the natural CRISPR-Cas, it has CRISPR RNA and also the transactivating CRISPR RNA or tracer RNA. And these two will come together and then form a complex with Cas9. But then the guide RNA that we're using in the lab right now is the combination of these two structures combined into one smaller structure for even easier use. And this one is called gRNA. The paper was published by Dr. Charpentier and Dr. Doudna group, GNEC, yeah. It was published in 2012. Very, very beautiful paper. I wish my result was like that. So beautiful paper. And yeah, and as some of you might have known that the natural CRISPR is actually the bacterial immune system. But how does it work exactly? And the purpose that I introduced this slide is because later there will be some of the wording, some of the, the word that will be used when if you students read the paper about CRISPR. Yeah, some of the things like protospacer or the PAM sequence or something like that. Yeah, so what are these things, right? Yeah, so when this is the bacterial cells, and when it's infected by the bacteriophage virus, its unique sequence of the phage virus called protospacer will be incorporated into the genome of the bacteria. And this spacer is flanked by repeated sequence. And all of these clusters are, yes, this is the CRISPR region in the genome of the bacteria. And when the bacteria gets infected by the same virus again, it's going to get recognized by the guide RNA. And this complex is going to cleave the genome of the bacteria. So the protospacer basically means the sequence of the target region that we would like to target. And the PAM sequence is the protospacer adjacent motif, or is the three nucleotides that the Cas9 will recognize. And yes, so this is how the engineered CRISPR-Cas9 cleaves the DNA and cause double-stranded break. in very similar fashion. Okay, now you start to understand how CRISPR-Cas9 are discovered and how it works. So I'd like to introduce you with the expanded toolbox of the CRISPR. The first one that we have been talking about is the Cas9. And as of today, you might have known so many Cases right now, right? Cas12, Cas13, yeah, CPF1, yes, but let's focus on the Cas9 for now. For the Cas9, there are quite variation to it. The first one is the regular Cas9, the conventional Cas9 that cause the double-stranded break. And if you look at the structure over here, you can see that there are actually two endonucleases sites. The first one is H and H, and the other one is RUVC. And as you probably can see here that the second modification of Cas9 is the Cas9 nicases, or the nCas9. It's simply, if you mutate and make one of the endonucleases site dysfunctional, then you, instead of getting the double-stranded break, you're going to get like the single-stranded break, or the nicking. So this is like the nicases. And the last one, if both of the endonucleases sites are mutated and can no longer function, this is the last, it will give rise to the last variation, which is the date Cas9 or dCas9. So what is the modification further of the first version? So the first version, as everyone might have known, that the off-target effects of CRISPR-Cas9 nucleases resulting from the unintended binding and cleavage. So researchers have been, trying to utilize the combination between rational design and selection to develop high-fidelity Cas variants. Yes, so there's so many things that you can buy now. Yes, students, yes. And this is not very surprising that all of the Cas9 that are available through the company, they have the paper. So you might want to check the paper before you decide, right? Yeah, which one you want to pick. And then we can also mitigate the off-target effects by optimizing the guide RNA using the example of these two freely available online software. And then the second one is nicases. And I think the nicases is very trending right now because the nicase can be used as the base editors or the prime editors. If the nCas9 is tethered with deaminase enzyme, for example, with the cysteine deaminase enzyme, it can cause the C to T transition, while if it's tethered to adenosine deaminase enzyme, it can cause A to G transition. But as you can see that the base editor is limited to just transition, which is just the mutation within the same base types. There's also the prime editors where the nCas9 is fused to the RT and used together with the PEG RNA to allow the transversion and even further incorporation of shorter sequence. And the last one is dCas9. Yes, because these are not causing any double-stranded break, so it's not going to cause directly cause the conversion of the DNA sequence in the genome, but it can be used to regulate the expression level by tethering it to some certain transcription activator or transcription repressor, or even the epigenetic modifier enzyme, like the TET or the DNMT. And this is a very busy slide, but I would like you to look at just a few points on here that after the identification of Cas9 in 2013, they also, in 2015 actually, they also discovered a Cas12a and Cas13. So what are these? This slide shows the two different classes of Cas. The difference between these two is only, okay. The first one is like, the first class is multiple protein together to become endonuclease, while the class 2, you only need one protein as the endonuclease. And Cas9, of course, belong to class 2, and this is Cas12a, as previously known as CPF1. It could result in a double-stranded DNA break, and the Cas12f has much smaller Cas protein, and the Cas13 is a little special over here because it doesn't actually cause a double-stranded DNA break, but it cuts the single-stranded RNA the same way as Argonaute protein does. Okay, so now we know all the tools, right? So we're ready to do some gene editing, but what could possibly go wrong? Yeah, so because the pitfall of CRISPR-Cas9, as everyone has heard before repeatedly, that the big problem is the off-target gene editing. And the reason is because the proto-spacer sequence is only nucleotide, 20 nucleotide long, and that's about the same length as primer. Yeah, so when you do PCR, sometimes you get the non-specific amplification. So that's actually the same principle as to why CRISPR-Cas9 sometimes bind to some other places that we don't want it. But there's some way that we can avoid this. So if the mismatch happens at the seed sequence, which is around 7 to 12 base pair proximal to PAM sequence, is not going to induce a double-stranded break. But if the mismatch is more distal towards the 5' end of the sgRNA nucleotides, it may result in double-stranded break. And so after we do the CRISPR-Cas9 with our cells, how are we going to detect the off-target activity? So these are, the first generation, second generation, and third generation. I, at the beginning of my training PhD, around not so long ago, 7 years ago, we were still using this first generation. It's just amazing how fast this field evolved in a very, very short time. And this illustration gives you a very nice idea as to how they come up with the new approach. And I think this last one is, most of the paper came out in 2015. It gives you a really nice, unbiased way to detect the off-target gene. For example, if you decided to go with the guide-seq, the way that they use it is that they insert a specific sequence into the break site, and then they do the whole genome sequencing looking for the integration of that specific sequence, and that's how they identify the off-target double-stranded break. So now after we're done with the gene editing with CRISPR, this is what I usually do. So first of all, I have to remember that the diseases are actually caused by abnormal protein, whether it be quantity or quality. So even after we do the alteration in the DNA sequence, we will have to check if the final proteins really answer our question, and that's the first thing. So yeah, the first question that I usually ask myself is, what is the consequence of the resulting mutation? Is it silent or is it consequential? And if it has the consequence, is that the consequence that we're looking for? And then the second one is the genotype is actually very important too. Is the edited gene has heterozygous edited genotype or homozygous or compound heterozygous to begin with. And then lastly, we ask about the off-target frequency and potentials, consequences of such mutations. Yes, and this is just to remind all the students all of the possibilities that could follow the double-stranded break. And this is some of the consequences that follow the point mutation. You can see that we can get all the possibilities of phenotypes. So please keep in mind what you're getting. Yeah. And also the interaction between the two alleles are very important too, because that dictates the phenotype. Okay, so now gene therapy in current medical practice. I have 2 minutes for 2 bullet points. I'll do it. Okay, yes. So this is very quickly because we have revisited this quite multiple times. So as of now, since the first gene therapy product came out, 27 gene therapy products have been approved worldwide, with 8 were the CAR T cells against CD19 BCMA antigens. 2 are exclusively approved in China. So for the US FDA approved, we have total of 6. And yes, this is the first of the second worldwide gene therapy approved product. And the first time that I look at this table, the first thing that I was looking for was, where's Thailand? And unfortunately, we don't have any gene therapy approved product yet, but as Ajahn Jatuporn said earlier this morning, that we're come together here because that's our mission. Yes. And another thing that, yes, and this is the 2 products that were exclusively approved in China. And the other thing that I was looking at this table is that CAR T cells really dominates the approved products. So it's trending. We're in the right place. And the next question is, those are the approved products, but what about the things that are in the pipeline? Meaning it's the drugs that are still in the development. Again, CAR dominates the pipeline. 40% of the pipeline, of the drug development pipeline, is CAR T, and the rest are just others and some other kinds of immune cells. And when we break this group down, you can see that most of them are still focusing on oncology. And the last one, CRISPR technologies in CAR T cells. And this is very short slides summarizing the potentials of CAR T cells, of CRISPR technologies in CAR T cells. And because of the characteristic of CRISPR, that we can specifically knock out or expressing desired DNA at a very specific locus, we can use this to knock out or re-express any kind of molecules that we want. For example, if we knock out the B2M, which is the part of the MHC class 1 and TCR, we can use this as the part of universal CAR T, allowing the allogeneic transplantation of CAR T. And we can also do the knock-in using the CRISPR-Cas9 expressing CAR and knockout TCR at the same times. Or we can even knock out like these exhaustion markers. And so last night I was doing this, fun experiment. This is the last slide already. You're very close to lunch now. Yeah. Also bear with me because I'm very excited about this actually. Yeah. So I asked a very simple question. So what do the trends look like about, the gene editing methods and CAR T? So the methodology is to, go to the PubMed and then use the keywords. Something and chimeric antigen receptor. And the y-axis is the article counts. Yes, and the first one is CRISPR and chimeric antigen receptor, mesh terms. Yeah. And as you can see, that is really trending up for CRISPR. And then I also searched for talents, yes. And yeah, it's been very humble and yeah, and the trend hasn't been changing so much. And then for the transposon, it's actually going up, yeah. But we have a lot of transposon specialists here, yes. And I can see that transposon has specific situation that, yeah, when we want to bring the cost of the CAR production down, yeah, it's very, very useful that way. So you're going to meet with a lot of transposon specialists. And for the lentivirus, it's also going up. And the retrovirus was popular before, but it's kind of going down a little bit recently because it's being replaced by lentivirus. Yeah, so that's like the short summary of everything. And yeah, I hope you get something from the lecture. And thank you very much for your attention.